what are microbubbles in ultrasound imaging? Microbubbles are extremely small, gas-filled spheres, typically ranging from 1 to 10 micrometers in diameter, that serve as contrast agents during ultrasound imaging. They create a significant difference in acoustic impedance compared to surrounding tissues, allowing ultrasound systems to capture detailed images of structures like blood vessels. For example, when a patient is injected with microbubbles, an ultrasound scan of the heart becomes much clearer because these bubbles strongly reflect the sound waves. Imagine a dark room where everything blends together. Then, sprinkle in tiny glowing beads that instantly highlight the edges of objects. These beads are like microbubbles, making important structures stand out. How is the gas core chosen and why is it important? The gas core is the inner part of the microbubble and is usually made from inert gases such as perfluorocarbons or simple air. These gases are chosen for their low solubility in blood, which ensures that the microbubbles remain stable long enough to travel to the desired imaging site. For instance, perfluorocarbon is frequently used because it does not dissolve quickly in the bloodstream, maintaining the bubble structure during the examination. Think of it as inflating a balloon with a special gas that doesn't leak easily, so the balloon stays inflated longer. A common question is, why can't we just use regular air? Regular air dissolves too rapidly in blood, whereas these special gases prolong the imaging window. What role does the shell play in a microbubble? Encasing the gas core is a thin shell made from lipids, proteins, or polymers. This shell is essential because it prevents the gas from diffusing out too quickly and helps control the bubble's mechanical properties like elasticity and strength. A typical example involves lipid-based shells, which are favored because they mimic the natural cell membranes found in the human body. One might ask, why not leave the bubble uncoated? Without a protective shell, the gas would escape almost instantly, rendering the microbubble useless for imaging. Picture wrapping a delicate object in bubble wrap. Without it, the object would be damaged. But with it, the object is protected and secure. How do lipid-based shells enhance microbubble function? Lipid-based shells not only secure the gas core, but also allow the microbubble to oscillate effectively when struck by ultrasound waves, generating clear diagnostic signals. They are designed to be biocompatible and to interact favorably with the body's natural tissues. For example, lipid-coated microbubbles are widely used in cardiac imaging because their flexible, natural-like membranes lead to more predictable responses under ultrasound. You might wonder, what makes lipid shells better than synthetic alternatives? Their natural composition minimizes immune reactions, much like how hypoallergenic materials are chosen for sensitive skin. How are microbubbles generated through mechanical agitation? Mechanical agitation is one of the simplest methods to create microbubbles. In this process, a solution containing the gas and shell material is vigorously shaken or stirred, leading to the formation of tiny bubbles. In a laboratory, a vial containing a lipid solution and gas is mechanically agitated, similar to shaking a bottle of soda to release carbon dioxide bubbles. A common question is, does this method produce uniform bubbles? While effective, mechanical agitation often results in a wide range of bubble sizes, which can affect imaging quality. Think of it like shaking a snow globe. The particles scatter unevenly, creating a mix of sizes. What is sonication, and how does it help in microbubble formation? Sonication involves using high-frequency sound waves to induce the formation of microbubbles. When a lipid solution containing gas precursors is exposed to these sound waves, rapid pressure changes cause the formation and collapse of gas cavities, leading to the generation of microbubbles. For example, placing a sample in a sonication chamber causes rapid and efficient bubble production. You might ask, how is sonication different from mechanical agitation? Sonication generally produces a more controlled and rapid formation of microbubbles, much like using a high-powered blender that uniformly breaks down ingredients into fine droplets. What advantages do microfluidic techniques offer for microbubble synthesis? Microfluidic synthesis uses tiny channels on a microchip to precisely control the mixing of liquids and gases, resulting in microbubbles with very uniform sizes and properties. In a microfluidic device, the flow of a lipid solution and gas is carefully regulated, ensuring that each microbubble is nearly identical. A common question is, why is uniformity important? Uniform microbubbles provide consistent acoustic signals, leading to clearer and more reproducible ultrasound images. Imagine a high-tech assembly line where every product is made to the exact same specification. This is the advantage of microfluidics in creating reliable microbubbles. How is size and uniformity controlled? And why does it matter? The size and uniformity of microbubbles directly influence their resonance with ultrasound waves. If the sizes vary widely, some bubbles may oscillate too much while others barely respond, resulting in poor image quality. For instance, in an imaging scenario, a uniform batch of microbubbles provides predictable and enhanced contrast, making structures like tumors easier to detect. You might ask, what happens if the sizes are not controlled? It's similar to baking cookies. If every cookie is the same size, they bake evenly, but if they vary, some might burn while others remain undercooked. Uniformity in microbubbles ensures a consistent and reliable imaging outcome. What determines the resonance frequency of microbubbles? The resonance frequency is largely determined by the microbubble's size and the properties of its shell. When exposed to ultrasound, microbubbles vibrate at a specific frequency that maximizes the reflection of sound waves, enhancing image contrast. For example, microbubbles designed for liver imaging are tuned to resonate at frequencies optimal for distinguishing liver tissue from surrounding structures. A frequently asked question is, why is tuning the resonance frequency important? It's similar to tuning a musical instrument. If the instrument is perfectly tuned, it produces harmonious sound. Likewise, optimally tuned microbubbles generate clear and precise diagnostic signals. What is nonlinear oscillation in microbubbles and how does it enhance imaging? Nonlinear oscillation occurs when microbubbles vibrate in such a way that they emit not only the original ultrasound frequency, but also harmonic frequencies. These additional harmonics are then detected by ultrasound systems to create images with improved contrast and detail. For instance, harmonic imaging techniques exploit these extra signals to differentiate microbubble echoes from those of the surrounding tissues. You might ask, how does this benefit ultrasound imaging? Think of it like a piano that, when played, produces a rich array of overtones alongside the fundamental note, adding depth and clarity to the music. The harmonic signals from microbubbles enrich the overall imaging symphony. How do stability and longevity affect microbubble performance? The stability and longevity of microbubbles in the bloodstream are critical for ensuring they provide effective contrast during an ultrasound scan. The choice of shell material and gas type plays a significant role in how long the microbubbles last before dissolving or collapsing. Researchers often add stabilizing agents such as cholesterol to the shell to slow down gas diffusion. A common query is, what happens if microbubbles aren't stable enough? It would be like trying to use a soap bubble in a windy environment. The bubble would burst before it could serve its purpose. Stabilized microbubbles, however, can endure long enough to circulate through the target area and produce high-quality images. Why is biocompatibility a critical consideration? Since microbubbles are injected into the human body, all materials used in their construction must be biocompatible and non-toxic. Lipid-based microbubbles are preferred because their composition is similar to natural biological membranes, which minimizes the risk of immune reactions or toxicity. For example, extensive clinical studies have demonstrated that these microbubbles are safe for routine diagnostic use. A common question is, what if the materials aren't biocompatible? It would be like using harsh chemicals on sensitive skin, potentially causing irritation or allergic reactions. Ensuring biocompatibility makes microbubbles safe and effective for medical imaging. How can microbubbles be functionalized for targeted imaging? Functionalization involves attaching specific molecules, such as antibodies or ligands, to the microbubble's shell, enabling them to bind selectively to target cells or tissues. This targeting enhances the diagnostic capability of ultrasound imaging by highlighting areas of interest, such as tumors. For instance, microbubbles can be engineered to target cancer cells, making the tumor more visible during the scan. You might ask, how does this targeting work? Think of it like a delivery package with a built-in GPS system. It is programmed to find and attach to a specific address, ensuring precise delivery of its contents. Can microbubbles be used for therapy as well as imaging? Yes, microbubbles are increasingly being explored for their therapeutic potential in a field called Diagnostics, where they serve both diagnostic and therapeutic roles. In this application, therapeutic agents can be incorporated into the microbubble shell, which then delivers the drug directly to the targeted site upon activation by ultrasound. For example, in cancer treatment, microbubbles loaded with chemotherapy drugs can be directed to a tumor where ultrasound triggers localized drug release, minimizing side effects. A common question is, how is this different from conventional drug delivery? It's similar to a smart delivery drone that not only reaches the precise destination, but also releases its package at just the right moment, providing a highly targeted treatment. What does the future hold for microbubble technology? The future of microbubble technology is promising, with ongoing research aimed at enhancing their imaging and therapeutic capabilities. Innovations include the development of multifunctional microbubbles that combine improved stability, uniformity, targeted delivery, and real-time monitoring capabilities. Researchers are experimenting with new materials, advanced synthesis techniques, and novel functionalization strategies to overcome current limitations. A common question is, how will these advances change medical practice? Imagine the evolution of a simple multi-tool into a sophisticated all-in-one device. Future microbubbles are expected to revolutionize personalized medicine by offering simultaneous diagnosis and treatment in a minimally invasive manner. These advances promise a new era in medical imaging and targeted therapy, making procedures safer and more effective for patients.